Welcome to the first class of Probability and Statistics. During this first class, I will give you some general information about the course, followed by a general introduction to statistics. As you all know, statistics will be a year-long course. Everything you need for the course can be found on Eufora. This contains course notes, slides, all the exercises and the practicals. During the lecture class, I will explain the theory to you and I will place accents to what's important. I will try to explain you the concepts and the ideas of statistics, what to use, when to use it. But part of the course will remain self-study, so we will not cover all the details that are given in the written notes. Those you will have to study yourselves. The exercises are split in two parts. You have the written exercises, where you will manually solve statistical problems, and you have computer-based exercises, where you will do this with the language R. For the written exercises, the goal is that you are able to understand and verify statistical information so that you can perform a simple test manually, but most importantly, interpret the results and see if the test is valid, when the test has to be used, and so on. The computer-based exercises are in R, a statistical package which we will learn during the course. The goal is that you can solve statistical problems using R, but also interpret the computer output and perform simple data analysis. The email addresses below are of course mine, and then the second one is of the TA, which is Negin Harandi. For the exam, there is no partial examination, so the entire examination is at the end of the year. Examination will be an open book exam consisting of two parts. So because it's a year-long course, you will have 3 hours in the morning and 3 hours in the afternoon. In the morning, you will get a written examination of 3 hours. In the afternoon, a computer-based examination. Both parts can cover all theory and all exercises, and they are set up to test your insight and your logical reasoning. So you will be asked to perform simple tests, to do simple calculations. You are asked to analyze statistical output, perhaps read a paper and discuss the results in it. And both parts of the examination are obligatory. So the examination consists of two parts, but it is one whole. If you don't show up for one of the parts, you will never be able to succeed for the course. Next to the examination, there is also continuous evaluation. So during the year, you will get 3 group projects of 4 persons per group. This will test your ability to work with the statistical package R and to report your results correctly. You will have to take part in all 3 projects. They are an obligatory part of the exam, and you will not get partial exemptions for those. If the projects that you hand in are of bad quality, then you will get at most 7 out of 20 for the entire course. It is therefore important to do well on the projects, also because they form an integral part of the examination and they count for quite a lot of the marks. As you can see, the division of the marks is the following. There is 30% of the final marks on the written examination, 30% on the computer-based examination, and 40% is on the projects. So if you hand in decent or good projects, you already gained a lot of your final score. 20% is on project 1, testing, 10% on project 2 on regression, and 10% on project 3 on simulation. The goal of statistics is to discover general statements, laws or rules that are valid on a population level. How does this work in practice? In practice, you often start with some experiment. So usually a small scale experiment, which will give you an initial ID. So this will give you some results, and these results can give you a hypothesis or an idea of what might be going on. But the result that you obtained was on a very small experiment. And so basically you now want to check if this also holds on a larger scale. So is this result also valid on large scale? So we will be saying, is it valid on a population level? Is it valid for everyone? Some examples might be climate change and feeding the world. So you would research what genes are associated with drought stress in plants. So you would take a small sample, you would take some plants, you would put them in severe conditions, you would dehydrate them, and then you look at the genes and the ones that are, you're trying to find the ones that are associated with drought stress, And then you will repeat this experiment on a larger scale to see if it is generalizable, if it holds also in the population of plants. Aging successfully, find biomarkers to adapt medical treatment on a specific patient. So personalized medicine, how do you adapt the treatment of a patient to the patient itself? So you will have to look on the gene level, which medicines work better for a certain person than others. And so statistics can help get an idea about this, because it will try to generalize the thing you saw on one person to the entire population. So did the effect that I saw, was it because of chance that it happened in this person, or is it generally valid for everyone? So also in biosciences, which you are following, there's a lot of empirical research. There are huge amounts of data which are gathered, and you want to make sense of it. So you have these data, you have this huge amount of numbers, and you want to get useful information out of it. The scientific method can be split up in a number of steps. So we distinguish 7 steps. The first step is data and scientific knowledge. So data, in this case, preliminary. So preliminary data, which gives you an idea about what is happening, what hypothesis do I want to research, or you might have some idea, some scientific knowledge, something that you saw before that you want to investigate. With that, you will formulate a hypothesis, so you will make a statement about what you think is happening. And with this hypothesis, you will try to get something testable. So you will try to make a deduction from the hypothesis and reformulate it in something that can be tested with a statistical test. We will see the details later on. But once you made the deduction, then it's time to collect the data. Collect the data has to be done in a reproducible way, so someone else has to be able to reproduce the experiment that you are doing, to verify it, and then with these data you will test the hypothesis experimentally. You will test it in such a way, so you will collect the data in such a way that the effect that you are seeing can be generalized, so that it's not only valid for the sample that you collected, but that it is valid for the entire population, so for the entirety of all the objects that you are looking at. You will analyze your data, interpret results, you will draw conclusions. Then you will publish those results, and then often some other scientists will try to reproduce the result, especially when they have doubt about the validity. So where will statistics come in? Mostly in steps 4 and 5. So collecting the data, how to collect the data, how many data points do I need, and so on and so on. And then the analysis of the data, the result interpretation, and the conclusion. In practice, step 5 will also often be the start of refinement. Because you get more insight in the effect, it can happen that the hypothesis that you made was not specialized enough or not detailed enough, and you will be refining what you are looking at, and you will restart the cycle. You will redefine a hypothesis, you will redefine testable effects, and you will recollect data, and so on and so on. So basically, statistics is the science that revolves around collecting and analyzing data. So it will help you to determine correct experimental design, which is the next chapter. It will help you to learn from the data that you have collected. It will help you to learn to quantify and report the variability and uncertainty. So the first thing is the correct experimental design. So it means what design or how do I have to set up my experiment? How many subjects do I have to take? Where do I have to test? How do I have to test? What do I have to test? Then learning from the data means what statistical test will I apply? What can I get out of it? And what are the results I am seeing? And then the last part, the quantification of variability and uncertainty, is basically you take a sample from the population. So you are investigating something, but you can never look at the entire population. So if you want to test a drug, for example, you will not test the drug on the entire population. You will not give it to all people, but you will take a group of people. You will take, for example, 100 persons, and you will test the drug on that person. Because of the way you selected the sample, you get a certain result. If you select another sample, if you select other people, the effects might be slightly different, and so you get a different result. And so the question is, this uncertainty, this variability in the results that I am seeing because of the sample that I have chosen, how can I quantify it? What can I say about it? Is it significant or is it not significant? To introduce you to the several steps of the scientific method and statistics, we will be looking at the BBC documentary. The documentary is talking about homeopathy. So homeopathy basically is a form of medicine in which people believe that you can be cured using an active substance which is diluted over and over again. So you will also see this in the video, but basically what they do is they take an active substance, something that they think causes the problem, and then they dilute it and they dilute it and they dilute it over and over again until it is extremely diluted, and then they make sugar pills of it. And then they use those sugar pills as a medicine instead of regular drugs. So the question is, will it work? Some people are convinced it does and they swear by it and will never take regular drugs. Other people are very skeptical or just don't believe in it. So in the documentary, what you will see is that some scientists did research on homeopathy and he published a paper in Nature, so the most prestigious journal, saying that homeopathy really works. There were, of course, serious doubts and his claim would have to be checked. And so what happens next, you can see for yourself in the documentary. So go to Euphora, watch the documentary, and then come back to see the rest of this video clip. You can find the video next to the slides. So if you saw the video, you're now going back, you remember the first part of the video was about the research question. The research question being, in general, does homeopathy work? Or, to put it into their terms, does water have a memory? Can water remember the active substance? And then suddenly someone, Jacques Benveniste, comes up with the statement, it does work. So he had strange findings in his experiments and he couldn't explain those. And he came up with a new hypothesis, memory of water. So what he states is that water has a memory, so water can remember the effect of the molecules. And with that, so he had the hypothesis in step 2. In step 3, he then reformulates this hypothesis in something that can be tested. So he will say memory of water corresponds to the deduction if anti-IgE antibodies are strongly diluted, so they are diluted over and over and over again, and they are heavily shaken, which was an important part in homeopathy, then the information that those antibodies contain, they are transferred to the water. So you don't need the antibody anymore, but the water will remember the effect. And so it can be detected by an allergy test with modified basophils in extreme dilutions. So he changed the general hypothesis of water has a memory to something that he can actually test in his laboratory. Then he designs a new experiment, step 4, to test this memory of water hypothesis. So he sets up the experiment. He then performs the experiment. And from the experiment, he looks at the results, the outcomes, and these tell him that water indeed has a memory. So he claims that he has proven the fact that homeopathy is working. He then makes a paper out of it and he publishes it in Nature. So he distributes the results in Nature. The people at Nature, however, were very skeptical. So they had reservations with it, and they also put in an editorial reservation to make that clear. So they did publish the paper because they couldn't find anything wrong with it, but they made a reservation and they said, we need to test it independently, we have to verify the results. And so they composed a team to verify the results, consisting of John Maddox, the editor, who had received the paper, Walter Stewart, who is a scientist and a fraud buster, and then James Randi, who is a magician and a skeptic. And then we arrive at step 7, reproduction of the results. When the experiment was repeated, it seemed to confirm the hypothesis that homeopathy is working. However, something went wrong. The problem in this case was with the experimental design. What they noticed was that the researcher, the scientist who performed the experiment, knew which tubes contained the homeopathic water and which contained the normal water. And thus the scientist who performed the experiment favored the tubes who contained homeopathic water. Not necessarily intentionally, but it is a very human reaction to do. And so by doing this, a bias was introduced. There was a skew towards the positive side. They gave more effect to the homeopathic water than it should have had. To avoid such situations, blinding is introduced, or blinding can be helpful, basically meaning that the codes to the samples are given at random, and the person who's doing the experiment does not know these codes, so he only checks with the codes after the entire experiment has been performed. So he is looking at the results, but he has no clue beforehand which of the tubes contained normal water, which of the tubes contained homeopathic water. And so he cannot be biased, he cannot be influenced by the knowledge that he has. And so the more subjective the measurement is, the more important blinding is. You can even go a step further and do double blinding. Then both the test subject and the scientist do not know what treatment is given, which is standard procedure for any drug testing. So neither the patient nor the doctor knows if the drug that he is giving to the patient contains the actual active substance, so it's really a drug, or it is a placebo. That way, neither the doctor nor the patient can be influenced by the effect of it. The question then remains, why does homeopathy seem to work? Basically, it will have to do with the placebo effect. The fact that the patient is improving does not necessarily mean that the medicine is working, but it can be due to his brain thinking that it is working and thus fighting the illness or the affliction himself. Placebo-controlled studies are thus very important in biomedical sciences. So it means that if you test a drug, you will have a group with patients who receive the drug, and on the other hand, you will get a group of patients who receive a placebo, so a pill which looks very similar, identical, but does not have the active components. So the patient actually does not know if he received the drug or the placebo, and thus if you see an effect of the drug, you know that the effect is really due to the drug. Of course, you can then go into discussion about the ethical limitations of using a placebo. Why would you exclude some people from treatment? What if the drug really works and you don't give it to people? So these are ethical aspects, but they are not statistical aspects. So in statistics, we just look at, is it working or is it not working? The ethicals are not for us, but they are for some ethical committees to decide on. And even though Benveniste's claim was refuted and it was shown that it is not working, the controversy went on. The experiment was repeated using a flow cytometer, so an automatic counting machine, in the hope that this counting would be less subjective than the manual count. And the results showed again that homeopathy seemed to work. However, this test again was not blinded, and so if you rerun it again, it shows that it's not okay. So it's not blinded, it's again the same problem. The scientist knew which tube was containing what, and so there was a bias, there was an influence of the scientist performing the research on the outcome. And then Randy proposed the ultimate test providing $1 million to anyone who could prove anything physically or scientifically impossible. So you had the $1 million paranormal challenge, but unfortunately this challenge has been withdrawn and is no longer valid. And then Horizon tried to repeat the experiment. So they created 2 × 5 test tubes with a 5C dilution. 5 of them had an active ingredient, 5 of them had pure water. So the first one were corresponding to the homeopathic water. The other 5 were just plain H2O. The tubes were then randomly labeled, so it's a blind experiment. The scientist who will be performing the experiment, who will count the results, has no idea about which tube containing what, and after labeling, the dilution went up to 18C. After further dilution, the tubes were relabeled to prevent all fraud, and then human cells were added, and a flow cytometer was used to count the activated cells. The experiment was simultaneously performed in 2 independent labs to test on the one hand whether or not the result is reproducible, but also you get more information by having the experiment at 2 different places than in only one laboratory. And so each lab was told that there were 20 active and 20 placebo dilutions, just to avoid that all of the tubes were labeled as negative or as not active. So they knew that of the 40 tubes, I have to label 20 as positive and I have to label 20 as negative. And then the results they would send back would be compared to the actual substance in the tubes. The data were then analyzed by a statistician, starting with data exploration. So just a plot of how are the data distributed, which tubes contain an active substance according to the testing, which tubes contain just plain water. And next, a more detailed summary was made. The first lab had 9 tubes of the homeopathic dilution and 11 of the controls labeled as being negative, so labeled as being a control. You can then put those results in a contingency table, and on the lines you see there's a line for homeopathy, there's a line for control, so it means that here on the first line you have the true tubes containing homeopathic water, and out of those 9 were considered by the lab as being a control group and 11 were considered as being homeopathic water. So these 11, these are correctly labeled. We have 11 tubes out of 20 which are correctly labeled as homeopathic water. And then the same thing, because you had 9 negative in the homeopathy group, you will have 11 negative in the control group. So these are also correctly labeled. So for each group, 11 out of 20 have been correctly labeled. If the homeopathic water and the normal water would have the same effect, then you would assume, you would expect that the distribution is 10 versus 10. So you would expect 10 homeopathic waters in the negative group, 10 in the positive group. In this case, we see there is a slight deviation noticeable. So 11 out of 20 were correctly classified. So it's a little bit more than you expect if everything happens completely at random. But then the question is this an indication of a true effect? Is it really that the homeopathic water is working better than normal water, or is this just by chance? If we look at the lab of Wayne, so the second lab that performed the experiment, we see an opposite result. So for the homeopathy, 9 of them were correctly labeled and 11 were incorrectly labeled. And same thing for the control, 9 correct, 11 incorrect. So here we find the opposite of what the other lab had found. And if you repeat an experiment, there is seldom exactly the same result, because there is always a matter of randomness in the experiment that you are performing. Think, for example, of flipping a coin. So you flip a coin, you know that you have 2 sides to the coin, heads and tails, and theoretically, each of those has a 50% chance to land on top. So theoretically, if you flip a coin 100 times, you expect that 50 of those coin flips will give heads, 50 of those coin flips will give tails. If you then perform the experiment in reality, you will see that if you do 100 coin flips, the result is almost never 50-50. There will always be a deviation from this theoretical result. So even if there is no effect, by chance, your results can differ slightly from what you would expect theoretically. But the question is then, what is slightly? How far do we call a deviation slight? And when does the deviation become significant? Or, otherwise said, how many correct positive tests are needed to have sufficient evidence that the homeopathy is working, that there is a true effect of the D tubes? To do this, we rely on probability theory. So we need to find the probability to find at least X correct positive tests by pure chance if D and C do not differ. So if D and C do not differ, so it means if there is no effect in reality, so the homeopathic water and the true water are just the same, how many deviations can we get before it becomes too unlikely? So you do that using probabilities. If X is 10, we see that the probability is 62%. So it means that if there is no effect of homeopathy, then we have 62% chance to observe 10 correctly labeled tubes. If we observe 11 correctly labeled tubes, there's only 37% chance. For 12 it's 0.17, so 17%, and we see that it decreases very rapidly. So it is very unlikely to classify at least 15 samples correctly if there is no true effect. So we saw a probability of 0.02, so it's extremely small. So it's, if we see 15 samples which are correctly classified, then we are almost certain that there is a true effect, so that the effect of D and the effect of C are different. So observing 15 correctly labeled tubes would be a strong evidence in favor of D. However, we only observed 11 correctly labeled tubes, and so it's possible in 37% of all cases. So here there is absolutely no evidence that homeopathy has a significant effect, that it is actually really working. What could have gone wrong? Every experiment is subject to random variability, and so because the experiment is subject to this variability, the conclusions will also be variable. As an example of random variability, you might think about the fact that the same treatment can have different effects on different people depending on the location where they are living, their age, their weight, and so on. Furthermore, even if D and C are equivalent, by chance we can still observe that 15 tubes would have been correctly classified. So this can be expected in 2 out of 1000 experiments. The chance is very slim, but it is not zero, it does exist. So for such an experiment, we will incorrectly conclude that D is effective, while in reality it is not. Intuitively, we also know that we cannot make any statement on the population characteristics based on a finite sample with absolute certainty. So there is always a measure of uncertainty that you will have to report when reporting the results that you found. What can statistics be used for? It cannot be used to prove anything. If you can prove anything, then you are misapplying, misabusing statistics. It can be used to design an experiment and also to analyze data when there is uncertainty. You can quantify the uncertainty, you can try to control the uncertainty, and you will have to report that uncertainty. And why do we see statistics in our program? Because scientific literature is filled with statistical analysis and terminology. So if you go to the industry later on, if you continue your studies, if you go to research, you will always be confronted with scientific literature, and you will always be confronted with statistical testing, and you will have to be able to interpret, to critically read what is written there. So you will have to verify that the statements that they are making are true or not. So the main objective of the course is to give you the necessary tools to critically evaluate statistical information in scientific papers. Such a critical view is needed because a lot of statistical analysis are performed by scientists who have limited or no statistical knowledge, and so you have to be able to see that what they are writing is really true or whether or not they made a mistake somewhere. Also, should you enter research, then you will have to perform experiments. You will have to be able to interpret the results of those experiments. You will have to report them. You have to explain them to others. And you have to do that in a statistically sound and correct manner. So if you want to be successful in academics, then you have to have a decent statistical knowledge because the quality norms for statistical analysis are ever and ever increasing. So the secondary objective is that we will enable you to perform a simple data analysis yourself. So we will give you the tools to set up your experiment, to perform the experiment, and then also to interpret the results and then transmit your message to the rest of the scientific community.